Good job. You remember the code?
Normally I'd say tomorrow, but I don't know what you're like. I know, I'll come by. I'll come by at 5 today, and we can look at it. But the reason I want to get out of here as soon as possible, because I don't want to get stuck in the snow.
Would you mind doing it at 5 today?
All right. No later than 5.
Or I can do Monday as well, if you're here.
I'm here. Yeah, definitely. Yeah, why don't you come Monday?
Sorry.
Monday. 5 o'clock.
Actually, well, I'm excited for classes tomorrow.
All right. So come to my office today at 5.
I'll be really quick. Thank you.
Micro's going fast.
All right. I just wanted to do that.
All right, let's get started. All right, attention, everybody. At my last lecture on B cells, I forgot to put in a little unit, I realized right after class, on memory B cells. And the reason I forgot to put that unit in there on memory B cells, because not much is known about memory B cells. You remember memory T cells, we know a lot about, different types of them. You know, there's resident memory, there's peripheral memory, there's central memory, there's effector memory. And that's it for T cells. And that's mostly for CD8 cells. We assume CD4 cells have a similar phenotype distribution. But B cells, there's less known about what the different memory phenotypes are. So we mostly understand the following processes take place, mostly, is when B cells become activated in the germinal center, and remember back and forth between the light zone and the dark zone, and it starts first in the paracortex at the primary focus. What happens to them then? Right, so remember you get the distribution between memory B cells and plasma cells. I'm going to also tell you a little bit more about plasma cells. The memory B cells, most of the B cells which are produced during the initial immune response are killed off. That's called activation-induced cellular toxicity. And I'll tell you more about that today. But some of the B cells do become memory B cells. Now, if they become a memory B cell, for instance, in the gut, that's where they're going to stay. So if they encounter the antigen yet again, most likely they're going to encounter that antigen in the gut. So they're primed at the anatomical location to become activated right at the area where they first encountered the antigen. So that works pretty well. Some of these memory B cells might take up, however, residence in the spleen. So the spleen, as you know, gets all the microorganisms that infect our blood, and so it's not illogical that you would have these B cells residing in the spleen and then to be activated for attacks by follicular helper T cells there as well. That's a possibility. These B cells may also, just like T cells, circulate through the blood, go through the lymph system, and then back to the blood. And then when they encounter antigen again in the draining lymph node, they stay put, become reactivated, and then start producing plasma cells, and then a fresh batch of antibodies. So you can see, almost anything you could dream up in your mind, it probably goes on with memory B cells, but there's a lot unknown about this particular topic. So it's a rich area for research, and if you're interested in immunological research as an undergraduate or as a graduate student, it's a nice area to go into because so little is known about actually memory B cells as opposed to memory T cells. And even then, a lot we don't know, but memory B cells are particularly murky. And I also gave short shrift to plasma cells, because most people do, because they think, well, they're the final effector cell producing antibody, and they don't live very long, and why is anyone very much interested in them? Well, you should be, because they're the main source of antibodies, number one. Number two, for many years, people thought they were very short-lived, and after the immune response was over to the initial antigen, the plasma cells were killed off, and that was the end of it. But we know now that's not the case. And the reason it didn't make any sense anyway to say that is because if you look at someone who was infected as a child with measles, for example, decades later that child would still or that now adult, would have antibody titers against the measles virus. So where is that antibody coming all this time? It must come from long-lived cells, and obviously the answer to that is that long-lived plasma cell. And in fact, people now know so well that there are such things as long-lived plasma cells. They have their own acronym. It's called LLPC. So there you go. And LLPCs, long-lived plasma cells, reside, as I told you, mostly in the bone marrow, but also in the gut tissue, mucosal tissue, so that you're constantly producing antibody from these plasma cells, which can protect you in the gut. But in any case, in the bone marrow, they know a lot more about them. They receive positive signals from various cytokines and other factors to stay alive. And the plasma cells in the bone marrow, at least the ones which have been looked at, we now know they express molecules which inhibit apoptosis, specifically the caspase cascade. So we see molecules such as BCL2, et cetera, BCLX, a variety of them, which are upregulated in these long-lived plasma cells, and that's what keeps them going. They also receive messages for other transcription factors to be produced, again, by cytokines. One of these cytokines is called APRIL, kind of a nice name. I forgot what that acronym stands for, but at any rate, and in the bone marrow, the cells which are nurturing to the plasma cells are really weird. They're eosinophils and megakaryocytes. These are two cells you would never ever think of being nurturing or encouraging plasma cells to be long lived. Now, does anyone know what a megakaryocyte is? Yeah.
It produces platelets.
Right, produces platelets, exactly. So the last cell I would ever suspect would be involved in helping us produce antibody over periods of decades. It's just kind of an odd thing, but nonetheless, that's the case. And eosinophils as well. Less shocking, but nonetheless not totally intuitive. Eosinophils also help plasma cells live for a long time. So, and there's more things going on with plasma cells and the memory B cells, but I'll let that go for this course. Yeah.
Would you say that it's called when the most B cells that are produced in the first immune response are killed?
Yeah, they kill themselves off because.
Right, but what was that called?
Oh, I forgot what I call it. But at any rate, activation-induced. Here, I'm going to give it to you. AICD, activation-induced cell death. I think I called it cell cytotoxicity, but activation-induced cell death.
Thank you.
Okay, just one more acronym to chalk up. All right. So now we're going to talk about cytokines. I've been alluding to cytokines probably from the first day of this class, and now we're going to actually look at them as a group of molecules. But this is a very complex topic to talk about because many different cells produce many different types of cytokines, and we can't possibly learn them all. But we're going to look at a few of them and then how they are activated and what they actually do. And the first two slides are a list of different cytokines. And one does innate immunity and one is for adaptive immunity. Right, so let's look at the innate immunity. And innate because they're produced by cells which are part of the innate immune response. That's the cell source for these cytokines. And the adaptive ones are mostly produced by lymphocytes, hence that's why they're considered part of the adaptive immune response. Nothing too surprising there. Now, you may think this is not intellectually very exciting, but I think it is because of all the research that was involved in discerning what these various molecules do. And in your book, there is an appendix at the back. And one of the appendices, the second one, I think, lists these different cytokines, tells you the cell source, what the molecule does, and if you knocked it out, what would be the result on the host. So you may want to look at that and check that in your book, because it'll give you some idea of the complexity of this particular field. All right, the first molecule identified as a cytokine was actually discovered in 1948. That's the year I was born. for those who are all interested. So maybe that's why I was predetermined to become interested in immunology and inflammation. I don't know. And the molecule in 1948, this is on, was called endogenous pyrogen, EP, endogenous pyrogen. And the reason it was called endogenous pyrogen was it produces a fever. So this is a molecule that's produced endogenously in an organism, which leads to a febrile response. However, in the decade since 1948, people tried to get a more uniform naming system for cytokines, and instead of them getting trivial names based on what they do, they just gave them even worse names, which was interleukin and then a number. So most cytokines are called interleukin 1, 2, 3, 4, 5, etc., up to 30-something. And the problem with that type of nomenclature is you have no idea what the molecule does by its name. So you just have to sit there and memorize them, or if you would, carry that appendix with you and determine what the individual cytokines actually do. It's called interleukin because they thought at first that these particular molecules are produced by one type of white cell and interacted with or affected the behavior of an other white cell, hence the term interleukin rather than intraleukin. So it's between two different white cells, interleukin. That doesn't always hold up, so the name isn't quite accurate. In any case, we're stuck with this nomenclature forever now. All right, so interleukin 1, again, the first one discovered is produced by our old friends monocytes, macrophages, probably dendritic cells, and it's induced when TLRs bind to a particular PAMP. And we already talked about that right in the beginning of the course. And interleukin 1 is produced in a very interesting way. It depends upon a very complex series of steps called the inflammasome, which I also alluded to. And the inflammasome, if you may or may not remember, leads to the activation of a series of enzymes called caspases, but in this case, caspase 1. And interleukin 1 by itself, there doesn't seem there's any way for it to be secreted. It doesn't have the right amino end to look like it can be secreted. However, what happens is once you get the TLR engaged, that leads to the assembly of this very complex structure called the inflammasome, which then activates caspase 1, which then cleaves the amino end of interleukin 1 and allows it to be released. If carried far enough, the cell which is producing the interleukin 1, because of this inflammasome and caspase 1, can lead to necrosis of that particular cell. So the cell explodes. This isn't apoptosis, this is explosion. And releases the cytokine along with a lot of other materials you may not want. And this process has a new name. It's called pyroptosis. So we have pyroptosis along with necrosis and apoptosis. Apoptosis is controlled cell death. Necrosis is when the cell is being killed either because low oxygen levels or a cytotoxic T cell may lead to a necrotic state, although not usually. So necrosis cancer cells can die of necrosis. But pyroptosis is due to a TLR engagement, an inflammasome being produced, lots of caspase 1 being produced, and somehow that sends a signal for the cell to explode, literally explodes, and releases all these inflammatory cytokines into the circulation. And the reason people are interested in pyroptosis, is a little bit of an aside, but interesting, is that it's much involved with the spread of HIV. Because cells which are infected by HIV undergo the process of pyroptosis. And for example, CD4 cells, we wouldn't normally think of having an inflammasome, wouldn't normally think of this whole cascade taking place in a CD4 cell. Nonetheless, when these CD4 cells are infected by HIV, they undergo an explosive eruption, And that doesn't help the host, but it certainly helps the virus. Can you figure out how that helps the virus? You have this explosion by the CD4 infected HIV cells. How's that going to help the virus carry out its mission, if you will?
Spreads more viruses.
Spreads more viruses, exactly right. So, fortunately, ferroptosis is something to be concerned about in an individual and the spread of HIV to uninfected cells. So, something to think about. All right, let's get back to interleukin 1. Interleukin 1 causes fever. That's what it was first named, endogenous pyrogen. How it produces fever, if this were a seminar in physiology, I'd tell you, but it's not, so I'm not going to tell you. But it's actually a very interesting story, how interleukin 1 leads to a febrile state. And I'm retiring, so you're never going to know from me anyway. All right, but besides that, it also causes the blood vessels to dilate, become leaky, so it adds to the inflammatory state. It also causes the liver to produce acute phase proteins, such as, and you know 2 acute phase proteins. Do you remember what the 2 acute phase proteins are I told you about? One begins with C. C-reactive protein, CRP. Can anyone name another acute phase protein? MBL, mannose binding lectin. All right. So there we go. We're done with interleukin 1. We only have 35 or so to go. We're not going to do an interleukin right now. This is tumor necrosis factor. And what is tumor necrosis factor abbreviated as? Always TNF. And there's 2 types. There's TNF alpha and TNF beta. To make your life miserable, TNF beta is also called lymphotoxin alpha. So we'll just call them TNF alpha and beta. Alpha, however, is produced by macrophages as well as by T cells, by both. And tumor necrosis factor does very much the same thing as IL-1 does. Probably also causes fever. It certainly does an induction of acute phase proteins. But TNF is a very, very powerful inflammatory cytokine. So powerful that it's involved in many autoimmune diseases. And therefore, a monoclonal antibody can bind to TNF and neutralize its effects. And this is called Humira, which you may have heard of. People with colitis or Crohn's disease, Humira can interfere with TNF's inflammatory processes, and many people can make them symptom-free. Not everybody, unfortunately, but many people make them symptom-free. Also, TNF is very much involved in rheumatoid arthritis, and therefore Humira works very well for some people who have RA, or rheumatoid arthritis. Now, tumor necrosis factor used to be called cachexin, and there's a word called cachexia, and cachexia means a state where you look like you're dying of cancer. So people who are dying of cancer have cachexia. People who are dying in the last stages of cancer, they're losing their muscle mass. And not only lose their fat, they also lose their muscle mass. And the molecule which is very much involved in a cachectic state during cancer, late stages of cancer, is TNF. And therefore that can cause a cachexic-like situation, cachexia, which seems a maladaptive outcome of TNF, but not entirely. Because TNF, by its ability to cause skeletal muscles to start to resolve, sends those amino acids in excess to the liver, and the liver then converts that into acute phase proteins. So the TNF actually allows more acute phase proteins to be produced, because it causes skeletal muscles to be broken down. That gives you a high supply of amino acids, which then go to the liver, which then can be induced to convert those into acute phase proteins. Again, what are the 2 acute phase proteins you know about? C-reactive protein and mannose binding lectin. There's other ones as well, which we're not going to talk about. Questions?
Oh, no.
Okay. All right, so that's TNF and interleukin 1. Interleukin 12 is important because it can give us a phenotype of T cell called a T helper 1 cell. T helper 1 cells
are the ones which help cytotoxic T cells become active. So we'll learn about that later today and next week, or the week after. And interleukin-12 has other abilities as well, but effects as well, but we'll learn about them later. Interleukin-6 is very much like interleukin-1 and TNF. You can see. In fact, these three molecules, IL-1, TNF, and IL-6, they induce each other transcriptionally in translation, so they can lead to positive feedback for these three inflammatory molecules. Okay, TNF, IL-1, and IL-6, all upregulating each other. And next come the interferons. It turns out there are three classes of interferons. There's class 1, class 2, and class 3. Actually, it's called lambda, though. So I was just funning you and also listening to the fire engine. I just never do up. I don't know. Anyway, so the interferons interfere with viral replication. That's how they got their name. Now, before any of you were born, maybe before your parents were born, people were excited about. God, I hope you don't have a fire alarm in this building. It's too cold. All right. People were excited about interferon. They thought it was going to cure everything. They thought it was going to cure cancer. They thought it was going to cure every disease you could possibly think of. This is back in the 70s. And I know none of you were born in the 70s. Were any of your parents born in the 70s? They were? I'm not sure. In any case, back in the 70s, we couldn't, molecular biology wasn't very advanced, so you couldn't produce biological molecules very easily. So people tried to purify interferon alpha at the time, and they finally got a little bit of it, and they decided this was going to be the cure for cancer, it was going to be the cure for every disease known to humankind. It turned out they used it experimentally, and what it did is it made people sick, and gave them fevers, made them very nauseated, and didn't do much for cancer, so it was a big disappointment. And actually there was a whole documentary at the time called The Big If for interferon, and again, that was a big disappointment. But interferon can be used, by the way, to treat multiple sclerosis. But anyway, interferon family 1 is interferon alpha and beta, and what it does is it causes primarily many things, but it causes an increase in class 1 expression. Well, why would that be useful to fight a virus? Why would it be useful to have an increase in the class 1 expression? Yeah?
Viruses infect cells and make them produce viral peptides, which they express more class 1, they'll be showing on their surface.
Right, and so a cytotoxic T cell, which we know is class 1 restricted, there's more class 1 presentation on a virally infected cell, it makes it more susceptible to be killed by a cytotoxic T cell, exactly. So that's why these interferon class 1 are useful. Upregulates class 1 and class 2 for that matter. All right, now let's get to the cytokines of adaptive immunity. Interleukin-2 is a favorite of most people who are involved in the adaptive immune response because T helper cells produce interleukin-2. And interleukin-2 is called an autocrine. That means it's produced by a T cell and works on the T cell that produces it. So the T cell secretes it. It then lands on a receptor on that T cell and then leads to the outcome of what's going to happen to that T cell. It very much involved in T cell proliferation and other things as well. We're not going to talk about activation-induced cell death really too much here, so just T cell proliferation and other things as well. Interleukin-4 is produced by T helper T cells, also by mast cells, maybe eosinophils, maybe natural killer T cells. And interleukin-4 can give us IgE. I told you that already. And also can give us a phenotype of a T cell called T helper 2, which we will look at later today. Interleukin-5 is produced by T helper T cells, and that gives you eosinophil activation. Eosinophils can be very much involved in destroying
metazoan worms in your intestinal tract. So this is good for elimination of worms. Another molecule I like a lot is TGF, transforming growth factor beta. This is not TGIF, which I also like, but this is TGF, transforming growth factor beta. And this one is actually anti-inflammatory. Inhibits. And it also may be involved in isotypic switching to IgA and inhibits macrophages. So TGF-beta puts a lid on the inflammatory response. It can suppress it. And we're going to talk about this because TGF-beta is also involved in the production of regulatory T cells. So we'll see that today. And finally, here's another interferon. This is class 2 interferon, or type 2 interferon. This is called gamma interferon. It's only produced by T helper 1 cells, maybe natural killer cells to some extent, maybe some CD8 cells, mostly T helper 1 cells. And what it does is it causes macrophages to become more powerful antigen presenters, more powerful phagocytes, more powerful killers, and et cetera, et cetera. So interferon gamma is really important in upregulating the adaptive immune response, along with the innate as well. So it's a very important molecule. Then there's a whole third class called interferon lambda, which has, we're not going to talk about the third type, so forget it. All right, now, the structure of interleukins follow a similar pattern. Unlike all the molecules we've talked about, interleukins do have alpha helical structures. Okay, so these are alpha helical structures. They have loops and not very much beta-pleated strands. Obviously, not very many there. So, unlike the other molecules, like the T cell receptor, like the antibody molecules we talked about, we do have alpha helical components to the interleukins. This is just showing you interleukin 2, but many of them have similar type of structures. What you want to make of that or not. Now, for an interleukin or a cytokine to work, it has to bind to a receptor. And there are really two main types of receptors. There's the interleukin 1 family receptor and everything else. Make it quite easy. So it's interleukin 1 family receptors and then everything else. Now, the interleukin 1 family we're not going to talk about, but I alluded to it before, because it involves IRAC and MyD88. Remember I showed you some things about IRAC and MyD88 at the end of cell signaling? There's 2 slides I showed you, which involve the TLR signaling. So the IL-1 works through a signaling cascade very similar to the one I showed you, those 2 last slides on T cell signaling. Very similar to that. But we're not going to talk about the cell signaling in this case. But in any case, IL-1 receptors we are just going to overlook. The other cytokine receptors consist of 2 or more subunits. 2 or more subunits, 2 to 3, actually. And the one I'm showing you here is for interleukin 2. And the reason I'm showing you interleukin 2 is because it's so important in controlling, again, the activity of the immune response. And the interleukin 2 receptors has 3 subunits, alpha, beta, and gamma, sometimes called different names, but alpha, beta, and gamma. And what's the most interesting aspect of this is the alpha subunit. The alpha subunit is usually not expressed on resting T cells. It's only on an activated T cell that the alpha subunit will be expressed. And when you get alpha, beta, and beta, gamma usually are constitutively expressed. They're usually always there on a T cell. But once the T cell is activated, then you get the alpha subunit trimerizing with the beta and gamma subunit. And when that happens, the IL-2 binds with high affinity, and you get all the effects of interleukin 2 that can be carried out. One, again, being cell proliferation. Now, the alpha subunit is so important to our understanding of immunology that we give it a CD name. It's called CD25. And if you're an immunologist, and you want to separate activated T cells from quiescent T cells, what you would look for is CD25.
Because it's the activated T cells which will express the CD25, whereas the inactive or quiescent or resting T cells would not. So it's a way of distinguishing, again, an active T cell from a resting T cell. Now, how would you do that in the laboratory? What kind of techniques would you use to identify whether CD25 is around or not on a T cell?
Cytometry.
Yes, you use flow cytometry. And there's plenty of antibodies against CD25 to identify whether cells are CD25 positive or not. So this is another CD I want you to memorize before you leave this course in less than a month. CD25, I definitely want you to memorize. Again, it's the alpha subunit of interleukin-2 receptor. Interestingly enough, regulatory T cells always express the alpha subunit. Regulatory T cells always express the alpha subunit, and therefore they have a very high affinity to bind to interleukin-2, even if they haven't been activated. So we can use CD25 not only to distinguish activated T cells from inactive, but also to try to fish out regulatory T cells. You'll see that today. I'll tell you how we can use CD25 to fish out regulatory T cells. So, what's the CD of the day?
CD25.
All right. Now, except for the interleukin-1 family, which, by the way, includes interleukin-1, interleukin-18, and interleukin-33. Interleukin-1, interleukin-18, interleukin-33. Okay. Is this campus burning down? Did anyone have a test today that they pulled the fire alarm? That sometimes happens. All right. Interleukin-1, interleukin-18, interleukin-33. Are all part of the interleukin-1 family, and I spent my postdoc studying interleukin-1 and most of my younger career here doing interleukin-1, but we're going to skip it mostly. And instead, we're going to talk now about cytokine signaling, and the cytokine signaling is true for most of the cytokines with the exception of interleukin-1. Now, if you want terminology, interleukin-1 cytokines are called interleukin-1 cytokine family. Almost all the other interleukins or cytokines are called the interleukin-1 or cytokine class 1 family. Class 1 family. And the interferons are called the class 2 family. Okay, so there's like 3 divisions. That overarching divisions, the interleukin-1 family, the class 1 family, which is almost every cytokine, with the exception of the interferons, which are called class 3. So that's more naming for you. And now let's look at the interferon in this case. And interferon in our story here is going to bind to a receptor which has 2 subunits, alpha and beta. When interferon binds to those 2 subunits, it causes them to dimerize. And when it does so, no shock here, that activates a family of tyrosine kinases, which are called JAK kinases, J-A-K. Now, what does JAK stand for as an acronym? Normally, I don't want to give you every acronym, but I'm going to give you this one. JAK kinases stand for Janus. Janus kinases. Does anyone know Roman mythology? Who was Janus? Yes.
Two-faced Roman god.
Right, two-headed Roman, or two-faced, right? Two-faced Roman god. Not two-faced being, talking behind your back and saying nice things in front of you. But it has 2 different faces. Hence January, because it looks at the old year and looks at the new. But Janus is called that because it faces both the cytoplasmic inner leaflet and the cytoplasm. So hence, that's why it's called a JAK kinase. But another name for the JAK kinase, because people were getting sick of all these kinases, was just another kinase. I was getting to an immunology joke here. JAK kinase, I like to think it's just another kinase, because there's so many kinases that leave one dizzy. At any rate, when these subunits dimerize, they each have a JAK kinase associated with them,
And again, abutting the cell leaflet on the inside and also the cytoplasm. And that causes auto phosphorylation. So the JAK kinase on this subunit phosphorylates the JAK kinase on that one, and this one phosphorylates the JAK kinase on the other subunit. So they phosphorylate each other. It's autophosphorylation. Okay? Then, when these JAK kinases are themselves phosphorylated, they phosphorylate a second stream of molecules called stats. Just like statistics. Stat. And when the stats get phosphorylated, they can dimerize with each other. And now they act as a transcription factor, which goes into the nucleus and leads to the transcription of some particular gene of interest in immunology. It turns out there's a drug now used for RA, and it's called Xeljanz, and here's its tofactinib, which inhibits these kinases, JAK kinases, and therefore it inhibits the ability of TNF to have its effects, because TNF works for those particular JAK kinases. There you go. I don't know if you heard of Xeljanz or not, but it's advertised all the time on TV. All right. And some of these molecules, the cytokines, are themselves made up of dimers. And some of the dimers are shared with one cytokine and another. So you have IL-12 here and IL-23, for example. They both share a subunit called P40. And this was a problem in research years ago, because what they did is they knocked out P40 in mice to see what the effects of IL-12 were. And they found out there were more effects for IL-12 than they thought were going to take place. And the reason is because not only do they knock out the ability of IL-12 to dimerize and produce its effects, but it also turned out they blocked the ability of IL-23 to dimerize and produce its effects. And it turns out IL-23 can lead to the production of T helper 17 cells, which are involved in autoimmunity. And IL-12 actually leads to a completely different set of processes. So here was a big mistake in around 15 years ago in immunological research. They knocked out P40, and they thought all the effects from knocking out from P40 were due to IL-12. To their surprise, they found out that some of those effects were due actually to IL-23, not to IL-12. And it took a while to untangle that mess. And you can see IL-12 and IL-23 work through different stats, different signaling molecules, so transcription factors. So that was a case where the research they thought was clear-cut. Turned out it wasn't. Right, this is just a list of different JAKs and stats for different types of cytokines and which ones they are involved with. You will have to memorize this for the final exam. I say this all the time, so you know I'm lying, but if I were a cruel teacher, I would give you the list of cytokines and then you would tell me the JAKs and you would tell me the stats. Or if I were really a jerk, I would give you the stat and then I would ask you what the cytokine is. So, you want me to do that on the final? I'll be happy to do that because it'll be easy to grade. Well, how many people want to have that on the final exam? You? How many people don't want to have it on the final exam? All right. Anyway, I'm not going to do that. All right, description of different types of T lymphocytes. There are many different types of T lymphocytes. You only know about T helper 1, and I don't even know about that. You know about cytotoxic T cells, you know about T helper cells. These are the only two you know about, right? At this point, T helper cells and cytotoxic T cells, right? Well, it turns out there are different types of cytotoxic T cells, which we won't discuss in this class. We'll just assume they're all the same, but they're not. And we also assumed up to this point all T helper cells were the same, but they're not. Turns out there's lots of different T helper cells, and I've showed you this diagram before. Well, this diagram is just showing you we have this non-committed T cell, and depending upon what cytokines are produced, when it's developing, it will develop a particular phenotype. These cytokines can either come from an APC and or another cell which has a TLR or PRR on its surface, and it produces a particular type of cytokine. These are called polarizing cytokines. And here's the family chart. Okay. And let's look at all the ones there are. There are regulatory T cells. There are T helper 17 cells. There are T helper 2 cells. There are T helper 9 cells. There's follicular helper T cells. I have told you about that one. There's T helper 1 cells, and there's T helper 22 cells. Now you notice there's 7 different T helper cells here. We're going to talk about 5 of them. I'm not going to talk about T helper 22. I'm not going to talk about T helper 9. And you can tell which ones I'm going to talk about because I put asterisks by them. Okay, so we're not doing this one, and we're not doing this one, but we're doing the other 5. All right. We're going to talk about regulatory T cells and 17 cells at the end of this lecture, or next Tuesday. We're really going to talk mostly for the moment about T helper 1 and T helper 2 cells. And then we're going to talk about follicular helper cells, which we still don't know a lot about and exactly how they function, but we're going to talk about them briefly today as well. Let's look at T helper 1 and T helper 2. In order to get a T helper 1 cell, the master cytokine that switches you to a T helper 1 phenotype, if you are a T helper 0. T helper 0 means you're a freshly minted T cell, which is CD4 positive, but I don't know what subphenotype you're going to be. Turns out, if you're exposed mostly to IL-12, and less importantly, these other 2 cytokines, it's mostly exposed to IL-12, the T helper 1 cell will upregulate a transcription factor called T-bet, take on the phenotype of a T helper 1 cell, which is mostly gamma interferon. And this is what the function is of the T helper 1 cells, and this is what happens if it becomes too active. So disease state is the cell becomes too active. So T-bet is the transcription factor with T helper 1. IL-12 is the master switch to get T helper 1. IL-12 mostly comes from dendritic cells and macrophages, mostly macrophages. T helper 2 is mostly induced by the switch cytokine IL-4. And for many years, people didn't know where the IL-4 came from in this early stage of development of the T cell. But we now know it probably comes from mast cells or eosinophils. IL-4 does not come from a dendritic cell, does not come from a macrophage, does not come from an APC, unlike what's shown here. It comes from, again, an eosinophil or a mast cell most likely. And IL-4 induces the transcription factor GATA3. And GATA3 switches you to the phenotype of T helper 2. And the most common cytokine associated with T helper 2 is IL-4, which is kind of interesting. Because it took IL-4 to get a T helper 2 phenotype, and the most iconic cytokine associated with T helper 2 is IL-4 itself. So it's kind of almost a positive feedback situation here. And IL-4, along with IL-5, helps you produce IgE and gets rid of, activates eosinophils and helps you get rid of worm infestations. And then after I give you more details about T helper 1 and T helper 2, we're going to talk about T helper 17 and regulatory T cells. Follicular helper T cells we already discussed enough on Tuesday, so I don't have much to say about that, except that to get a FH phenotype, you really need IL-21. And that begs the question, where does IL-21 come from? Because IL-21 is the iconic cytokine from follicular helper T cells. IL-21 does not come from an APC. Does not come from an APC. So where in the world does the IL-21 come from to give you the FH phenotype initially? I'm not sure. A little bit of unknown there. But we know if we take T cells in culture and just pour IL-21 on them, you'll get the phenotype of an FH, because then they'll start producing IL-4 and IL-21. And then for an FH cell, IL-4 is produced true, but the most important cytokine for an FH cell is IL-21. All right. Let's look at T helper 1 and T helper 2 cells, though. All right. So this is, I already told you what happens. We get a type 1 response. And this is our dendritic cell. It's producing IL-12. That polarizes us to produce T-bet through various stat connections here. And we end up with a T helper 1 phenotype producing gamma interferon. Type 2, this slide is incorrect. It looks like the dendritic cell is producing IL-4. That's not the case. IL-4 is not the source. It does not come from dendritic cells. It comes probably from mast cells, eosinophils. So your book is wrong in this case. And that happens. But again, the master switch here is GATA3. But there's a question here, which you should be asking yourself intellectually. This one, and this one. This one, and this one. And do you know what that intellectual question is? What biases the dendritic cell to produce interleukin-12? And then what biases another situation in another microenvironment to give you interleukin-4? And no one knows. And for many years, people argued you can't, in a single lymph node, have both the T helper 1 and T helper 2 response. It has to be one or the other. That's no longer the case, to be true. You get both T helper 1 and T helper 2 from a single lymph node. But again, why in one microenvironment you have an excess of IL-12 and give you the T helper 1 phenotype, and why in another microenvironment you mostly have IL-4 and give you a T helper 2 phenotype isn't known. So that's something to also investigate. Now, how many people want to write an NIH research grant, R01? I've given you 2 today. The 2 are, how do you determine in the microenvironment whether or not you're going to be biased to T helper 1 or T helper 2? No one's been able to figure that out yet. The second NIH R01 grant you may want to write is, how can you tell what memory B cells are, and how can you differentiate memory from naive B cells? We're still not entirely sure of that. So there's lots of areas to do investigation from today's lecture. So I don't know the answer to that question. I wish I did. And I'm hopeful someone in this audience who has a young mind, is energetic and intelligent and creative and hardworking will solve some of these problems. Anyone want to go on into research in this room? Research? 2 people? All right. Everyone else wants to become a doctor, a dentist, or a vet? Is that right? All right. We need doctors. I'm not denying that. But how do you think doctors know what to do? They have to rely on the research that informs them what they do in their clinical situation. So you just don't give penicillin because you say someone told you to do it. How do they know how to do that? I mean, so you have to do research. And anyway, how many people have done research? Okay, how many people have done a 93 or a 94 in biology? How many people have done a 193 or 194 in biology? How many people are doing a 199, which is a thesis? Anyone doing a thesis? In any department in science? That is, I don't care about I don't care about humanities. All right, that's a joke. I do like humanities. All right, so someone asked me the question that happens every year. Well, how in the microenvironment do you determine it's going to be T helper 1 and T helper 2? And what would my response be to you? I won't use an expletive. Blank, no. All right, it just isn't no. This is a nice little cartoon. It's not even correct cartoon because it's not the dendritic cell that's the source of the IL-4, so it's not even correct. So I don't know. Now, however, there is crosstalk between T helper 1 and T helper 2 cells. Crosstalk between T helper 1 and T helper 2 cells. So study this, because I'm getting sick of talking and blabbing to you. And blah, blah, blah. Tell me what the crosstalk is. By the way, a flat thing on the end of a line like this, a vertical flat thing, means it's inhibitory. This means it's inhibitory.
An arrow means it's excitatory. Inhibitory, excitatory. NKT is natural killer T cells. I told you about them. They can recognize lipids.
Should there be crosstalk right now, or?
What? Cross here, you mean? Yeah, talk all you want. It's Thursday, it's going to be a snowstorm. You'll have an exam for a month. Chill out, relax, but not too much because I don't want you to fall asleep. So enjoy the lecture. Crosstalk.
Tag yourself on IL-4.
Yeah, so the regulation of TBET downregulates GATA3. You have both.
I don't know if it gets downregulated, it just stops.
IL-4 leads to more IL-4.
It's upregulating yeah, and so
Yes, what happens is you get the IL-4. The IL-4 gives you T helper 2 phenotype is the main iconic cytokine with T helper 2 and IL-4. So there's even more in that direction. It turns out, not shown here, that gamma interferon causes the APC to produce more IL-12. That's not shown. That isn't shown, is it? That also. So thank you. Why'd you ask that?
is there something that they agree on?
Because right, if you have IL-12, you have TBET, and so you don't have GATA. It also comes from different kinds of stuff. IL-4 comes from like that, and IL-12 comes from dendritic cells.
All right, what's the bottom line here, class? What's the bottom line? They each inhibit each other. So once you are going down a path of T helper 1 and T helper 2, it's irreversible because what happens at that point is once you start emerging with that phenotype, it actively discourages or inhibits the emergence of the opposite phenotype. And that's why people thought in a single lymph node you wouldn't be possible to have T helper 1 and T helper 2. But you can, because they may be isolated in different follicles, so you may actually have, you do actually have both types of cells emerging. So so what happens is you get IL-12. IL-12 gives you STAT4, which gives you TBET, which is a transcription factor, and TBET actually inhibits the expression of GATA3, so that's going to discourage T helper 2. TBET, though, leads to a transcription of interferon gamma. Okay, it inhibits the transcription of IL-4 and IL-5. T helper 2 is the opposite. You get GATA3, which inhibits TBET. That gives you IL-4 and IL-5, but it also actively inhibits the gamma interferon phenotype. The end result for T helper 2 is you get a lot of IL-4 as well as IL-10 and IL-13. And because you get more IL-4 from the T helper 2 cell, that goes back and pushes even more cells to the T helper 2 phenotype, so you get a positive feedback. So IL-4 is produced in high quantities by T helper 2. That causes other cells, naive T cells, uncommitted, non-committed T cells, to become T helper 2. TBET, you get mostly gamma interferon, and gamma interferon causes APCs to produce more interleukin 12. So here's your positive feedback for the T helper 1 as well. So you get T helper 1 producing gamma interferon, and also secretes these as well, but also gamma interferon. Gamma interferon then crosstalks with the APC, dendritic cell or macrophage, and you get more IL-12, which commits you even further to the T helper 1 phenotype. All right, look at this one. Go ahead and discuss this one. It repeats a lot from the previous slide, but it gives you some more information. So look at this one. Yes, you can go and talk. Crosstalk. Figure it out.
Yeah, so they interfere. So the interferon gamma goes back to activate, but it also and
Feedback there.
and it goes back to the
Positive
one.
IL-12.
But it
Interferon gives you
also
more IL-12.
inhibits the IL-4.
So this is IL-4 and IL-4. This is gamma interferon IL-12.
IL-12 pathway is just less direct.
Yeah.
Yes. Right. Yeah. The two different molecules, where it's IL-4 and IL-4.
It makes IL-4. So the IL-4 goes back.
All right. It's only a week from today that Thanksgiving takes place. That being the case, does someone want to come up here and give thanks that you now can actually read the slide and figure it out instead of me doing it? So you're probably sick of hearing my voice because I'm sick of hearing it. Anyone want to come up here and explain this slide? I know you're paying and I'm getting your money. That's my salary. But how about we reverse roles? I won't give you any money, but come up anyway and figure this out. How many people are graduating seniors? Okay. What's the tuition and everything else here? 70? 70. All right. By the way, do you always write to your parents and tell them how much you love them because they're paying $70,000 a year for you to go here? I hope you say thank you to your parents. I'm a parent, and you don't hear that very often, so it's nice to hear. When you become parents, you'll know how cruel children can be. Anyway, can anyone figure this out? My usual suspects not even raising your hand. Some people in this room signed up for my seminar. I could single you out because I know who you are because I looked at the class list. Oh God! All right. All right. Let me test you quickly. What's the master transcription factor for T helper 1? T-bet or GATA4? GATA3 rather. T-bet. Oops. My mistake. GATA3. Yes, T-bet. And T-bet's going to give us T helper 1. What's the iconic cytokine for T helper 1? Gamma what? Gamma what? Gamma what? Interferon. And gamma interferon acts in the positive feedback way because gamma interferon causes the APC to produce more what? IL-12, which gives you more T-bet, which gives you more T helper 1 phenotype. Okay, let's go down to the bottom of the figure here. Again, we don't know the source of IL-4. Give me a source of IL-4, the master cytokine here. What's the source of IL-4?
Mast cells and eosinophils.
Yeah. Where did I type up here? No, I didn't type up here. Right. All right, eosinophils, mast cells. Is it from an APC though? No. And remember, your book is incorrect. It shows that it is. It's incorrect. All right. And what's the master switch here? GATA3. GATA3 gives you a lot of different cytokines. IL-4 being the iconic one. IL-4 then acts in the positive feedback way because now you have more IL-4, which will push more naive T cells to the T helper 2 phenotype. So once you commit to T helper 1 or T helper 2, it's irreversible, really. You're going to be committed in that way because of the positive feedback responses that are taking place. Okay, again, what's the point of T helper 1? DTH. Remember DTH? T helper 1 is involved in DTH. It also helps cytotoxic T cells, not mentioned here, become active. Very much involved in macrophage activation. Less likely, this third bullet point is probably not correct. That's probably mostly a follicular helper T cell, not T helper 1 involved in this. Okay. T helper 2 is eosinophil activation and production of IgE, and also activation of mast cells and production of mast cells and eosinophils, and that is good for getting rid of worms. Okay, so, all right. By the way, with the T helper 2 response, you get rid of worms because of eosinophils can kill them, mast cells can kill them, plus it causes your goblet cells in your intestinal tract to produce more mucus. And what is the good of that? Because the increased mucus slides those worms out of there and you defecate them away. So the T helper 2 cell helps you. Trying to think of a polite way of saying this. Defecate them away and you flush them down the toilet. And that's why dogs get a lot of worms, because what do dogs do? Because they're not too smart. They're sniffing around and they eat feces. Stupid thing to do. But somehow they survived as a species for so many years. All right. Now, how many people are dog lovers out there? How many people are cat lovers? Are any people cat and dog lovers? How many people don't like pets? All right. I won't tell you which of those three ones I am, but I'm definitely one of them. All right, anyway. I'm not telling you. All right, I want you to look at this and talk about this one. And I know in my evaluations, I get this every year. People say, it goes off in tangents which have nothing to do with immunology or physiology. That's, of course, I'm teaching. I know that, but I'm entertaining myself. Because I've heard all this before. All right, and also trying to wake you up. You all look so somnolent. You know what a somnambulist is? A somnambulist is a sleepwalker, and a lot of you. All right, anyway, go ahead. Figure this out. There's also an opera. Most of the slides from here on are going to be experimental, cartoons followed by real data. We're coming to some real data slides.
There was a study recently, a few years ago, doing toxoplasma gondii, depression,
Is that right?
depression, and I forget what else.
It's a pretty nasty thing. Pretty terrible. Next one's if you're a little leishmania, the next one's pretty bad too. All right, let me ask you this, class. Mighty 88. Remember I told you TLRs very often signal through IRAC, which is interleukin-1 receptor kinase, and Mighty 88 is involved in that as well. Mighty 88 is involved in TLR signaling, but also interleukin-1 family signaling, which I told you about at the beginning of today's lecture. And again, the interleukins, which are in that family, are interleukin-1, interleukin-8, and interleukin-33. But it's also involved in TLRs, and you can't produce interleukin-12 if you're missing Mighty 88. All right? So one of my favorite acronyms, Mighty 88, I just like it, the name. All right, so what happened to our wild-type mouse versus our knockout mouse? And the knockout mouse didn't have Mighty 88, so therefore it can't produce plasma IL-2, IL-12 rather, and it can't produce gamma interferon. And therefore, if you're the knockout mouse, do you survive this particular infection? No. So this particular infection requires a T helper 1 or T helper 2 response. T helper 1 or T helper 2 response?
T helper 1.
T helper 1. By the way, lots of research was done determining what type of microbe is more susceptible to T helper 1 versus T helper 2, but by doing these knockout experiments, either knocking out the ability to generate T helper 1 or knocking out the ability to generate T helper 2. How would you knock out the ability to have a T helper 2 response? What, if you were the researcher, what gene would you knock out? Right, IL-4, exactly right. If you knock out IL-4 and you get a certain outcome for a particular microbe, you know either T helper 2 is responsible for helping you get rid of that microbe or not. And that's how we know which particular cell type or T cell phenotype is involved in a particular disease process. Look at the next one. See what you can figure out for this one. Leishmania is a nasty, do not ever get this, Leishmania is a nasty, nasty disease. Can I say that again? It's a nasty disease which will torture you, literally. It's a torturous disease to have.
What happens?
Pain, eruptions, brain problems. Is that enough? All right. It's transmitted by an insect.
Insect?
Insect.
So I don't like bugs. Precisely.
At any rate, I'm not interested in your bug allergy, but let's go. What's going on? Read what's going on here. There's something evolutionarily here to tell you as well about the protozoans versus the mammal, the mouse. It's an interesting Darwinian story here. It's an interesting Darwinian story here.
Why is it a stick?
Oh, no. It's fake that anyway. So. But the outcome is true. How do you know that?
Emergency.
All right. Tell me the moral to the story in this case. If you have Leishmania, what do you want to be, T helper 1 or T helper 2 response?
T helper 1.
T helper 1. But the untreated mouse, what response did the untreated mouse present? T helper 2, and it died. Now, who's this helping? One would think it would probably help the protozoan because it can replicate and replicate and replicate, and as it gets more insects bite the person or the animal, which is infected, there's a chance for the protozoan to spread to other organisms. So it fools our immune system and biases towards a T helper 2 response. That's how the organism is allowed to allow more offspring to develop. Whereas with the T helper 1 response, you kill that Leishmania, and there's fewer offspring for this protozoan, and therefore, in a Darwinian way, it's less successful, obviously. So our immune system might be clever, but microbes may be more clever, because now it's, again, biasing our immune response to go for the T helper 2 phenotype rather than the T helper 1 phenotype. So that begs the question, how is Leishmania causing your immune system to be biased towards the T helper 1, T helper 2 phenotype, presumably because it's inducing the production of IL-4. That's what one would assume. So again, not by the T cells, but again, the innate cells, such as eosinophils, etc. All right. Now we're going to look at regulatory T cells and helper 17 cells. And on the left are the emergence of the regulatory T cells, and on the right are the emergence of helper 17 cells. Go read this and contemplate what the two divergent pathways are, two different pathways are for the emergence of regulatory T cells and T17 cells. I'll just say T17, it's easier, T17 cells. The master switch for regulatory T cells is FOXP3, and I want you to memorize that immediately. FOXP3 is the master transcription factor for regulatory T cell phenotype for peripheral regulatory T cells, not for thymic, but for peripheral regulatory T cells. We know very little about thymic regulatory T cells. Most of our knowledge is peripheral induced regulatory T cells, so FOXP3 for peripheral regulatory T cells. And ROR gamma T is a transcription factor for T17. And by the way, if retinoic acid, which is vitamin A, binds to a cell, it will prevent the transcription factor ROR from being expressed. All right, so go ahead and read this. I like this particular cytokine too. Hint, IL-23. I underlined it.
I'm using R8 to differentiate neurons.
Hmm?
I use R8 to differentiate neurons.
Sorry, what?
I use retinoic acid
Oh,
to
you did? Okay.
differentiate neurons right now.
This one inhibits T17. Is interesting. T17 cells might be, oh, never mind, I'm not going to blab it. All right, class, how do you get a regulatory T cell? What are the cytokines which induce, what are the cytokines which induce regulatory T cell? What is the cytokine which induces a regulatory T cell? TGF-beta. TGF-beta is the master cytokine in giving you the phenotype of the regulatory T cell. It's mostly in the periphery, or maybe only in the periphery. We're not sure what goes on in the thymus. So these are peripheral regulatory T cells. Keep that in mind. All these discussions about peripheral regulatory T cells. We know less about the thymic ones. Okay, and TGF-beta. In the naive CD4 cells exposed to that, we already know how, by the way, how we get a CD4 cell. Single positive CD4, that was on the second exam, so you know that. And then you end up with FOXP3 and the classic characteristics. If you were to do a two-color analysis on a flow cytometry of a regulatory T cell in the periphery, two things you would look at, FOXP3 positive, and what's the second molecule you would look for if you're doing a two-color analysis of a regulatory T cell? FOXP3 would be the other one? CD25, right. So you look for CD25 and FOXP3 positive cells in the periphery, in the upper right quadrant. That would be probably highly suggestive, but not entirely foolproof, that those are regulatory T cells. Not absolutely necessarily the case, but good indication that those are regulatory T cells. Okay, how do you make a T17 cell? How do you make a T17 cell? Not only do you need TGF-beta, but you also need IL-6 and perhaps IL-23. Again, I like this particular cytokine, so let's throw it in for sure. And that gives you the T17 phenotype through this particular transcription factor, which we know can be inhibited by massive doses of vitamin A or retinoic acid. Now, regulatory T cells downregulate the immune response. We want to get rid of them in cancer patients because regulatory T cells suppress the anti-tumor immune response. So you want to get rid of regulatory T cells in cancer patients. That's not really done commonly at this point. And T17 are involved in some autoimmune diseases, maybe involved in asthma. So we certainly want to know how to either downregulate or upregulate these particular cells. But interestingly enough, if you downregulate T17 cells, and you can do that with certain types of monoclonal antibodies to treat certain types of diseases like psoriatic arthritis. However, if you do that, one of the side effects of downregulating T17, unexpected, was to get an increased incidence of colitis and or Crohn's disease, inflammatory bowel disease. So even though T17 cells are associated with inflammation, if you downregulate them, for some reason, you get an upregulation in inflammatory bowel disease. All right, but to get T17, you need TGF-beta and you get T17. And T17 cells produce IL-17, and IL-17 activates neutrophils, is activates the inflammatory process on blood vessels, so it's very pro-inflammatory. Again, but why, if you suppress it, do you get a worse inflammatory bowel disease? It's not what one would expect, nonetheless does happen. Okay, now I was going to give you these, because these are fun slides, flow cytometry slides, but I don't have enough time, so we'll have to do that after this weekend, because I have to give you back your exams. And the average on this test was around 90.